Hello doctor, I am Shruq Sayed Ahmed and these are my teammates Amira Muhammad, Iman Abdusattar and Fatma Jalal, and our presentation is going to be about directed evolution of zinc finger nucleases. This is our content. So in the last years of 1970s decade, Michael Smith and his colleagues demonstrated that gene sequence could be altered by synthetic nucleotides, which results in changes in primary structure of any protein. He proved that this approach could be successfully implemented, opening the way to a new research field that has become subsequently known as protein engineering. For his pioneering work in this field, he was awarded Nobel Prize for Chemistry in 1993. The definition of protein engineering: the design of new enzyme or protein with new or desirable function, and it's based on the use of recombinant DNA technology to change amino acid sequences. Our model is zinc finger nucleases, which consist of protein subunits, each subunit attached to zinc metal ion. So in action, the zinc finger protein subunits gather and bind to an endonuclease enzyme called FOC1, which is then dimerized to bind to the double-stranded DNA and cause a double-strand break. This allows any other foreign DNA sequence to be integrated inside the DNA genome. The directed evolution of FOC1 cleavage domain could be achieved by one of these two approaches. The first one is error-prone PCR, which is used to introduce diversity into the catalytic domain of FOC1 by changing the concentration of different metal ions like magnesium and manganese and nucleotide mixes from DNTPs to DITP. The second approach is the DNA shuffling to combine beneficial mutations, starting by a gene pool with related DNA sequences, and after fragmentation, a random DNA fragments are produced, which are reassembled to form library of recombinants that is then selected or being screened to choose the mutant gene encoding protein with improved properties. After that, the phase of selection is achieved by cloning the zinc finger nuclease library into a plasmid called PDAS, electroporated inside an E. coli cells to allow protein expression to choose the best version of the protein. The resulted model is called Sharkey variant, which represents the most efficient FOC1 cleavage domain. Variant consists of two amino acid mutations, S418P and K441E. Together, they enhance the cleavage capability and site specification by 10 folds and 5 folds respectively, relative to the activity of wild type of the cleavage domain of FOC1. There are so many applications for the zinc finger nucleases, as for example, custom designed zinc finger nucleases offer a way to deliver a site-specific double-strand breaks to the genome. This makes targeted gene correction or gene editing a viable option in human cells, which can treat so many genetic disorders. This was our review about one protein, but obviously there are so many other proteins that may have better versions to be discovered. Thank you.